Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties of the enzyme in the course Enzyme Science and Technology. So far what we have discussed about how you can be able to produce the enzyme by cloning the enzyme into a suitable vector. Followed by we have also discussed about the different types of methods and approaches, how you can be able to purify the enzyme. And then we have also discussed how you can be able to perform the enzyme assays. And subsequent to that, in the previous module, we have also discussed about how you can be able to utilize the enzyme assays to design the different types of inhibitors. And when we were discussing about the enzyme inhibitor approaches, we have discussed about the traditional approaches where you are actually going to screen the compounds and you may not need the structure of the enzyme or the inhibitor, and you will be able to get the inhibitor to inhibit the enzyme. And then we also discuss about the targeted approach, where we have discussed about the ligand-based approach, receptor-based approach, or the computer-aided inhibitor design. So, if you have designed an inhibitor, you would like to understand the mechanism of its action. You would like to understand how the enzyme is inhibiting the protein enzyme, and how these inhibitors can, because once you know the mechanism of the enzyme inhibition, you can be able to work on that mechanism and you can be able to improve the activity of those inhibitors. So in today's lecture, we are going to discuss about the how you can be able to study the inhibition of the enzyme and how you can be able to do the different methods to understand whether the enzyme inhibition is reversible or irreversible and all other kind of aspects. So in this lecture, we are going to cover that particular aspect. So as you can say that the enzyme inhibition, so once you have developed the inhibitor, right, you have developed a inhibitor, it is actually going to block the enzyme, and that's how the enzyme will not be able to convert the substrate into the product, right? Now this inhibitor could be of multiple types. So what is the inhibitor? Inhibitor is a chemical agent which is inhibiting or poisoning the enzyme, which means it is actually blocking the enzymes in such a way that the enzyme will not be able to convert the substrate into the product. Depending on the type of inhibitions, the inhibition could be of irreversible inhibitions or the reversible inhibitions. Irreversible inhibition means it is going to be a permanent type of inhibition. Once you add the inhibitor, it is actually going to block the enzyme, and then this is going to be a irreversible process. Whereas in the case of reversible inhibition, the enzyme inhibitor will actually going to inhibit the enzyme, but if you do some treatment or if you do some other method, it is actually going to reduce, or it is actually going to relieve the inhibitions. So the first question comes, how you will be able, so suppose you have designed an inhibitor, right? And you have tested that onto the enzyme, right? So suppose you have tested it on the enzyme and you will find that it is actually forming a complex with the enzyme, and because of that the enzyme is unable, so when the enzyme is active, it is getting converted the substrate into the product, but when the enzyme is inhibited, it is not being able to convert the substrate into the product, right? So it will not be able to convert the substrate into the product. But this information is not complete until you know whether the inhibition is reversible or the irreversible. So the first thing what you have to understand is whether the inhibition is reversible or the irreversible. So how we can actually be able to use the different types of methods to understand this. So the enzyme, so inhibitor could be reversible or irreversible, right? Now, In an irreversible enzyme inhibitor, the enzyme inhibitor is actually going to interact with the enzyme, and that's how it is actually going to make a permanent complex, which means it is going to be a permanently lock the enzyme into this particular conformation. Whereas in the case of reversible inhibition, what will happen is that you have an enzyme, and when it is actually going to interact with the inhibitor, and you have an inhibitor, when it will interact with the enzyme, it is actually going to form the enzyme inhibition complex, but that complex is being mediated mostly by the known covalent interactions such as hydrogen bonding, van der Waals forces, and electrostatic interactions. So this is reversible, which means if you add, for example, if this is being mediated by the salt bed interactions, you can actually be able to change the pH, and if you change the pH, this inhibitor is not going to be bind the in enzyme, or even if the inhibitor is bound to the enzyme, it is actually going to be changed. The other thing what you can also do is you can actually be able to do the other kinds of treatment, and that's how the enzyme inhibitor complex can be broken down, and that's how the enzyme can be reactivated its activity. So how there are many methods what you can actually be able to use to understand whether the inhibition is reversible or the irreversible. First thing what you have to do is, first method is you can actually be able to do the dialysis. Okay, so what you can do is, and I'm sure you all know about dialysis, right? So what we do is we take the enzyme, okay, and then you add the inhibitor, okay, and take this whole thing. For example, if I take the 0.2 ml of enzyme, and then if I add the 0.1 ml of inhibitor, okay, and then you add the buffer, right? For example, you add the buffer of 0.2 ml, okay. So this means this is the total 0.5 ml enzyme inhibitor mixture, okay. Now what from this 0.5 ml, what you can do is you can just take out the 50 microliter and you check whether the enzyme is active, right? That will confirm that the inhibitor is working and inhibitor has inhibited the enzyme. So it's not active, right? So that's why the enzyme is not active, right? So in active or inactive, right? So if the enzyme is inactive, you will know that the inhibitor is working. Now what you can do is just take this and put it into a dialysis bag. Okay, so you put the dialysis clips and put it into a dialysis bag. Okay, so you take this 0.5 ml and put it into dialysis bag, right, and put it into a beaker, right, with a lot of buffer, right. So you can put like for example 2-liter buffer. So what will happen is that when you put it into the 2-liter buffer, if it is a reversible inhibition, right, because you have increased the volume, the enzyme inhibitor complex is actually going to be because you know that enzyme inhibition complex is having the equilibrium with enzyme and inhibitor, right. So if you increase the volume, the equilibrium will go into this direction, okay, and because of that the enzyme is actually going to be get dissociated from the inhibitor, because the concentration of this complex is going to be lower down, and as a result it is actually will go into this direction, and as soon as it will go into this direction, the inhibitor will actually going to be removed from the inhibitor from the enzyme. And when the inhibitor is going to be removed, and since the inhibitor is of a small molecular weight, mostly the inhibitors are in the range of 500 Dalton, right? They will come out. So inhibitor will come out and will present in the outer environment, okay? And because of that, the enzyme, you are going to get a free enzyme. So enzyme will remain inside and the inhibitor will come outside, and that's how the enzyme. And now what you can do is just after, for example, if you do this for 12 hours, okay, and after that what you can do is just take out the enzyme and ask the same question whether the enzyme is active or inactive. And if the enzyme is active, okay, then the inhibition is reversible. If the enzyme is still inactive, okay, if its enzyme is inactive, then you will know that the inhibition is irreversible, because the inhibitor enzyme complex is not being mediated by the reversible forces. It is being mediated by the irreversible forces such as covalent bond and all those kind of things. So in that cases, the enzyme is not, inhibitor will not get going to be dissociated from the enzyme, and that's how you can be able to know this. So one method is dialysis. You can actually be able to do dialysis. The second, which is still the same similar method, but it is more refined. So what you can do is you can do the gel filtration. And I'm sure still you remember when we were talking about the gel filtration chromatography, that gel filtration chromatography is actually going to resolve the molecules based on the size. So what will happen is that you again make a mixture of 0.5 ml, and then you load this mixture onto a gel filtration chromatography. So what will happen is on the gel filtration, this 0.5 is actually going to be loaded onto a 25 ml of column. Okay. So because you have loaded into a 25 ml column, the complex is again going to be get diluted, and as a result, the enzyme inhibition complex is again going to be dissociated into the enzyme and inhibitor, and as a result, what will happen is that inhibitor is going to be separated from the enzyme. So what will happen is you will going to see the two peaks. You are going to see a peak which is for the enzyme, and you will going to see another peak which is for the inhibitor. So this is for the peak for the inhibitor, this is the peak for the enzyme when you run it on the gel filtration chromatography. So now what you can do is just take this whole peak, you can collect, and you are going to get the enzyme, and that you can actually again ask the same question, whether the enzyme is active or whether it is inactive. If it is active, then the inhibition is reversible, right? Because you have removed the inhibitor and you can be able to remove the inhibitor. If it is inactive, then you can be able to say that the inhibition is irreversible, because you still have the you have collected the enzyme fraction, but still the inhibitor is present, because inhibitor is of a small molecular weight, so it will be get separated from the enzyme. So this is the two method what you can actually be able to use very nicely. The third method what you can do also is you can actually do the dilution method. So what the dilution method is? The dilution method will say, I'll try here. Okay, so dilution method. Now, what you can do is in the dilution method, suppose an inhibitor is active at 1 micromolar concentration. Okay, so suppose the inhibitor is active at 1 micromolar, but it is not active. It is not active if you convert the enzyme, if you change the concentration at 0.1 nanomolar. Okay, this means if you do a dilutions, it will not going to be active. So at this time it is active, the inhibitor is active, which means it is actually going to inhibit the enzyme, but this concentration it is not active because it is very low, right? You will going to have. So what you can do is you can just make the reaction mixture, right? This reaction mixture, right? And you keep the inhibitor at 1 micromolar. Okay. Now you take the aliquot of the enzyme and ask the question whether the enzyme is active or inactive. So in this particular concentration, the enzyme is going to be active. Now what you can do is you just take 1 microliter of this, okay, and put it into 1 ml of buffer. This means you have actually done the 1000% dilution. Okay, so when you do the 1000 times dilution, what will happen is the inhibitor is 1 micromolar here. The reaction mixture is actually going to have the inhibitor, which is now in the 1 nanomolar, right? Because you have diluted it by the 1000 times. Now, in this again, you take the sum amount and you take the enzyme from here and you do the activity. So, if it is active, then you will say that the inhibition is reversible. If it is still inactive, right? If you got the enzyme which is inactive, then you will say that it is a irreversible. So, dilution method is not confirmatory result, okay, because sometimes what happen is when you dilute the buffers to 1000 folds or something, the enzyme its quantity also is going to be reduced, and in that case it's possible that the enzyme may not be very active. So dilution method is being done once you have the confirmatory test from the dialysis as well as the gel filtration chromatography. So dilution method is a quick method because it is just takes lot of time, because dialysis will take 12 hours, gel filtration chromatography also will take some time, but dilution method is going to just, you have to set up the reactions, take the 1 microliter, put it into 1 ml, mix it, and then you take the small aliquot and check whether the enzyme is active or inactive. If the enzyme is active, the inhibition is reversible. If the enzyme is inactive, then the inhibition is irreversible. So dilution method is a quick method, but it is having its own drawback that sometime when you do a 1000 times dilution or 100 times dilution, the enzyme concentration also will be very low, and at that particular concentration, you may not get a very good readable count. So these are the some of the analytical method what you can actually be able to use to study whether the type of inhibitions. Once you know the type of inhibitions, the type of inhibition could be reversible or the irreversible. So we have the irreversible inhibitions where the enzyme is actually going to make a complex with an inhibitor, and this complex is going to be mediated by the irreversible forces such as covalent bond and other kinds of interactions. So this is not going to be get dissociated. Whereas in the case of reversible inhibitions, the enzyme is going to interact with the inhibition and it is actually going to be reversible. So this enzyme inhibition complex is going to be mediated by the reversible forces such as salt bridge interactions. It is going to be mediated by the electrostatic interactions, and it is going to be mediated by the hydrogen bonding and so on. So because these interactions can be broken down, the inhibitor can actually get broken down and give you the free enzyme and inhibitor again. So in the reversible category, you have 3 different types of inhibitions. You can have the competitive inhibition, you can have non-competitive inhibition, and you can have the uncompetitive inhibition. So first we're going to discuss about the irreversible inhibitions, and then we are going to discuss about the reversible inhibitions. So irreversible inhibitor or irreversible inhibition, as the name suggests, it is actually going to be permanent, and it is not going to be get affected whether you dilute the enzyme, whether you do gel filtration chromatography, whether you dialysis, anything. Once you add the inhibitor, it is actually going to make a complex with the enzyme, and that's how it is actually going to destroy the activity. So irreversible inhibitor destroyed a functional group on the enzyme which is necessary for the catalytic activity. For example, diisopropyl fluorophosphate or DFP, which inhibits acetylcholine esterase. Okay. Another example is iodoacetamide. Iodoacetamide inhibits the OH group of serine and SH group of cysteine and imidazole group of the histidine. So they are mostly the irreversible inhibitor are actually going to go and bind or destroy these functional groups onto the enzyme, and that is going to be irreversible damage, and that's how the enzyme will not be able to recover its activity even if the inhibitor is present or not present around. There are some examples of irreversible inhibition of the enzyme. For example, diisopropyl fluorophosphate DFP, which inhibits the acetylcholine esterase, and serine proteases such as trypsin, chymotrypsin, and elastase. Then we have the British anti-leviside, which is an antidote for the heavy metal toxicity. Then we have cyanide, right? You remember that the classical case of cyanide, that cyanide, nobody know the taste of cyanide because cyanide is very toxic poison, right? And why it is poison? Because it is actually having a irreversible inhibition of cytochrome oxidases, and cytochrome oxidase is a very important component of electron transport chain. So if the electron transport chain is going to be blocked by a molecule, it will actually stop the production of the ATP. So there will be a no production of ATP, and as soon as there will be a no production of ATP, it is actually going to stop the physiological processes, right? And that's why the cyanide is going to be very potent, right? Inhibitor, right? Because it's going to be irreversible, and this because it cannot be allow the enzyme to be reactivated or something. That's why any kind of poisoning or toxicity by the cyanide cannot be reversed. Then we have the fluoride. So fluoride actually inhibits the enolase, and it removes the magnesium and manganese from the active side of the enzyme, and that's how it is actually going to deprive the enzyme from the crucial cofactors, and because of that, the enzyme will not be able to active. So in a typical reaction, what will happen is that this is a reaction where we are actually discussing about how the enzyme is actually how this DFP is actually inhibiting, right? So this is actually diisopropyl fluorophosphate. This is difluorophosphate, and what you see here is this is the fluorine, what is present onto this phosphate, right? And this is the DFP, right? And this is the enzyme which actually contains the active site serine, okay? And it has a serine OH, right? So you see the lone pair of electron here, which is present onto the oxygen of the serine. So what happens is that when it interacts, the fluorine is actually going to be removed, right? And this fluorine is going to be removed because its fluorine is going to be attacked onto the lone pair of the oxygen, and as a result, it is O is actually going to make a complex with phosphate. And now what you see here is that lone pair is also gone and does not have the functionally active OH group, right? And this functionally active OH group is very important because once the OH is present, it actually can take up the electrons from the substrate. It actually can give the electrons to the substrate, and that's how the functionality of the enzyme is solely being depend on to this particular functional group. So when you added the DFP, what DFP will do is this fluorine is actually going to attack onto this particular oxygen, and as a result, it is actually going to make a permanent complex. See, this is a covalent bond, right? So if it is making a covalent bond, whether you dilute this enzyme, whether you dialyze this enzyme, or whether you run it as in the gel filtration, this bond is not going to be broken down, and that's why this enzyme will no longer be functional. So this is actually going to be a non-functional enzyme because the inhibitor has blocked one of the crucial amino acid, and this crucial amino acid could be present in the active site, and that's how it is participating into the reaction. So this enzyme is functional enzyme, this enzyme is a non-functional enzyme. So this is the more or less the classical activity or classical way in which the irreversible inhibitors are actually inhibiting the enzyme. Now, irreversible inhibition of the acetylcholine esterase by DFP, right? So DFP is a very potent inhibitor. So what will happen is that when it is actually going to inhibit the acetylcholine esterase, it is actually going to interfere into the neural transport, and because of that, it is actually going to stop the communication between the muscle cell and as well as the neurons, okay? And as a result, it is actually going to stop the activity of the muscle cell, because you know that the muscle cells require the neural signal for the and it's their actions, right? If you want to know more about the muscle action, what you can do is you can actually be able to go through with some of my course of which is called as the basics of biology, where I have discussed about the muscular system, and I have also discussed about the nervous system. Okay, so how the acetylcholinesterase and the neural transmission works, you can actually be able to go through with these lectures, and they will actually going to give you the full idea. And once this is actually going to be blocked, the muscles will not actually going to be contract or relax and all those things, and that's how the person is no longer be able to move its body. Okay. So this is what exactly going to happen. The acetylcholinesterase will have the functional A active serine, and when DFP is going to be added, it is actually going to make a complex, and that's how it is actually going to produce the inactive enzyme. And this inactive enzyme is actually going to block the the transmission of the signal from the nerve cells to the muscle cell, and that's how it is actually going to stop the muscle contractions, and as well as, and so the person is going to be get paralyzed. So cholinesterase, this is exactly the same what we have shown, that you are actually going to have the RVT101 inhibitor, and that also blocks another receptor which is called as the 5-HTX receptor, which actually promotes the release of the choline and other neurotransmitters. And once you are actually going to block this particular receptor, it is actually going to stop the secretion of the neurotransmitters such as glutamate, acetylcholine, and all that. And because of that, it is actually going to stop the neural transmission between the two particular neurons. And same the other drug which is called as donepezil, and donepezil inhibits the cholinesterase, preventing the breakdown of the acetylcholine. And how it is actually going to be working, because it's going to stop the neural transmissions, it is actually going to increase the concentration of the acetylcholine and other neurotransmitters, leading to the improved cognitions. So irreversible inhibition of the enzyme with the SH group. So this is another example where the iodoacetamide can be used, and iodoacetamide is going to block the enzyme which has the SH group, which means it is going to block the enzyme where you have the functionally active cysteine. So enzyme with SH group such as the cysteine, when they are interacting with the iodoacetamide, enzymes are getting the chemically modified, and that's how the enzyme is getting converted into the inactive enzyme. So what happens is that you have the enzyme, for example, some enzyme which actually contains the active site cysteine, right? So it's going to have this, right, CH2SH. So this is the CH2, this is SH, right? And when you add the inhibitor you are going to add the iodoacetamide. So iodoacetamide is going to have the iodine, and SH is also again going to have the electrons, right? So this iodine is actually going to attack onto these sulfur groups, right? And as a result, there will be a covalent bond which is going to be formed between the sulfur and this carbon, right? And iodine is actually going to be removed, right? So Hi is actually, so this is going to be removed here, right? Iodine is going to be removed, and hydrogen is also going to be removed from here, right? And then there will be a covalent bond. Once a covalent bond is formed, this enzyme is going to be get converted into a inactive enzyme. So these are some of the examples where you are actually going to chemically modify the enzyme. Then we have the another variation of the irreversible inhibition, which is called as the suicide inhibitions. So suicide inhibitions is, as the name suggests, here when you add the inhibitor, the enzyme will actually going to go for the suicide, which means it is actually going to enzyme it's actually going to process the inhibitor. Enzyme is actually, so inhibitor is not irreversible inhibitor. It is actually, once it is going to be processed by the enzyme, it is actually going to be get converted into the irreversible inhibitor, and that's how the enzyme is actually responsible for converting a inhibitor into a suicidal inhibitor. So this is a specialized form of irreversible inhibition. It's also known as the mechanism-based inactivations, right? Because here the enzyme is going to be actively participate into the inhibition mechanisms. Enzyme inhibitor makes use of the enzyme's own reaction mechanism to inactivate it. Inhibitor, which is a structural analog, is converted into a more effective inhibitor with the help of the enzyme to be inhibited. So enzyme is literally committing suicide because they utilize the normal enzyme reaction mechanism to inactivate the enzyme. We'll take the example, then you will understand how what it mean actually. So what it says is that enzyme is actually going to process the inhibitor, and as a result is it actually going to be get converted into, it's actually going to process these inhibitors. So inhibitor is actually going to be go and bind to the enzyme, and then it is going to be processed, and that's how the enzyme is inhibitor is going to get converted into a inhibitor star. And then as soon as the inhibitor star is going to be formed, it is actually going to attack on the enzyme, and that's how it is actually going to form a enzyme inhibitor star complex. This is actually a irreversible. Okay, so this is actually going to be irreversible inhibition. This means the enzyme is itself acting on the inhibitor and making it on its own death actually, and that's why it is called as a suicide inhibition. So what is the properties of the suicide inhibition? It is going to be irreversible inhibitions. It is a very effective inhibitor compared to even the irreversible inhibitions where you have the covalent bond. It is synthesized with the help of the enzymes, which means the enzyme is actually going to be convert the inhibitor into a suicidal inhibitor, and then this suicidal inhibitor is actually going to act onto the enzyme. Examples are inhibition of xanthine oxidase by the allopurinol, which is a treatment of the gout. Then we have the alloxanthine synthesized by the xanthine oxidase using the allopurinol is more potent inhibitor of enzyme than the allopurinol. Then we have another example of arachidonic acid getting converted into a prostaglandin, which is going to be catalyzed by an enzyme which is called a cyclooxygenase, and it is inhibited by the aspirin. Aspirin is an anti-inflammatory drug, right? And then we also have an example of 5-fluorouracil. So 5-fluorouracil is getting converted into 5-fluorodeoxyuridine, and this 5-fluorodeoxyuridine is a more potent suicidal inhibitor because it is going to inhibit the thioredoxy synthesis, and it's going to inhibit the nucleotide synthesis, and it is being used in the cancer treatment. So 5-fluorouracil is a anti-cancer drug. So let's take an example of some of these mechanisms. So first example is allopurinol. So allopurinol is a drug for the gout, right? Gout, you know that gout is a disease of the joints, right? So the anti-gout drug is a suicidal irreversible inhibitor, mechanism-based inhibitor of the enzyme xanthine oxidase that works as the oxidase or the dehydrogenase. The enzyme commits suicide by activating allopurinol into a transition state analog, oxypurinol, that binds very tightly to the molybdenum sulfide complex at the active site. The molybdenum sulfide complex binds the substrate and transfer the electron required for the oxidation reaction. So what happens is that in a normal reactions, what is the xanthine oxidase is doing is it is processing the hypoxanthine to xanthine, and then from xanthine oxidase is getting converted into the uric acid, and this uric acid is actually a problem because uric acid is a metabolic byproduct. Remember that when we were talking about the amino acid metabolism, we'll say that there are so many nitrogen derivatives and other things are going to be produced. So you are going to produce uric acid, urea, and all other kinds of derivatives. So uric acid is actually going to be get deposited into the joints, right? And that's how it is actually going to cause the disease which is called as gout. And mostly it is a, sometimes it is a genetic disease and other kinds of things, right? So uh, now what happen is that when you have the allopurinol, right? So allopurinol is a inhibitor of xanthine oxidase, but when the allopurinol is actually going to be processed by the xanthine oxidase, it is actually going to be get converted into oxypurinol. Okay, and oxypurinol is a very potent inhibitor because it is actually going to bind the molybdenum sulfur center what is present inside the xanthine oxidase. So initially the allopurinol does not have any activity or any kind of binding to the molybdenum sulfur center, and that's why the allopurinol actually will not be able to inhibit the xanthine oxidase if the xanthine oxidase will not process the allopurinol to a oxypurinol. So allopurinol is getting converted into oxypurinol, and oxypurinol is actually attacking onto this molybdenum sulfur center and making a complex. And once it's making a complex, the molybdenum sulfur center is actually the active center which is involved into the electron transport or electron movement between from the enzyme to the substrate. And because of that, it will actually going to stop this in this particular all these reactions, which means it is actually going to stop the synthesis of the uric acid, and as a result it is actually going to give you the treatment for the gout. So this is what exactly happened. Allopurinol getting converted into the oxypurinol, and oxypurinol it's actually going to have this nitrogen, right? And this nitrogen is actually going to interact with the molybdenum and sulfur center what is present onto the xanthine oxidase, and that's how it is going to make the irreversible complexes. And because it is going to make a irreversible complex, molybdenum sulfur centers are no longer, or molybdenum sulfide center will no longer be able to transfer the electron from the substrate to the other molecules, and as a result they will not be able to catalyze these reactions. Then we have the mechanism of action of the 5-fluorouracils. So 5-fluorouracil is getting converted into a fluorodeoxyuridine, and that is actually a suicide inhibitor. So this is a carbon cycle. This is the carbon one carbon cycle, right? Through which the nucleotide and as well as the amino acid components are actually exchanging the carbon pool between the. So you'll see that these are the protein carbon pool, and this is the the nucleotide carbon pool. So when there is a shortage of carbon into the protein pool, the protein can actually take the carbon from the nucleotides. When there will be a shortage of carbon in the nucleotide pool, then they will actually do the reverse. And these are the some of the enzyme what actually going to function. So you have the dihydrofolate reductase, you have the thymidylate synthase, and all other kinds of things, right? And at this place you also have another enzyme which is called as the SHMT, right? So which actually going to convert the serine to glycine, and that's how the carbon, the CH3, is actually going to be transferred onto the tetrahydrofolate and form the N5-N10 methylene tetrahydrofolate. And then N5-N10 methylene tetrahydrofolate, which actually carries the carbon from the serine, is actually going to be taken up by this system, and that's how it's actually going to form the dihydrofolate, and then dihydrofolate is actually going to be converted into tetrahydrofolate and so on. So thymidylate synthase is a very active enzyme, and it is responsible for the nucleic acid synthesis, right? Because it is actually going to be involved into the synthesis of nucleotides, and that's how it's actually going to be. So if you thymidylate synthase, when it processes the 5-fluorouracil, it gets converted into fluorodeoxyuridine, and fluorodeoxyuridine is a suicidal inhibitor for the thymidylate synthase, and that's how it is actually going to block the thymidylate synthase, and that's how it is actually going to stop the reaction here. This means there will be no exchange of carbon pool between the protein chain and as well as the nucleotide, and as a result the nucleotide synthesis is going to be stopped, and that's how it is actually going to be blocked. The other enzyme, which is dihydrofolate, is actually going to be inhibited by the aminopterin and the methotrexate. These are the two other inhibitors, but these are not irreversible inhibitor. These are the drugs, right? 5-fluorouracil, what people take for the cancer, right? So this is a cancer drug. So apart from the cancer, we have another example, the therapeutic usage of the enzyme inhibitors. You can have the the clinical use for the epilepsy, so where you are actually inhibiting the enzyme which is called as gamma transminase, and the inhibitor is called as gamma vinyl GABA, right? Then we have the drugs for the antidepressants, which is called as MAO, and the inhibitor is the triclopyr phenylzal. These are the two different types of inhibitors. And then we have the anti-hypersensitive drugs, which is acetylcholinesterase. The drug's name is cabotripil and the inaprolart. Then we have the cardiac disorders. So there are so many ATPases, and then you also have the cardiac glycosidase. Uh, then we have a gout. So gout, your enzyme is xanthine oxidase. The drug is allopurinol. And then we have ulcers, right? So you can have many types of ATPases, because these ATPases are involved in the the proton transport and all that. So you can actually be able to use the omeprazole, right? And this is very popular, right? Omeprazole, pentaperazoles, and all other kinds of azole derivatives which you can use. And these are the all are irreversible inhibitors for this class of enzymes. Now, let's see the case study where we are actually going to study how you can be able to study suicidal inhibition kinetics, and how you can be able to understand the different types of the parameters and all that. So the case study is very simple, right? Where we are actually going to study about how you can be able to suicide inhibit, right? This is a suicidal inhibitor where we are actually trying to inhibit, where we are trying to study inhibitor which is called as NAC. Okay, so NAC's full form is N-acetylcysteine. Okay, and it is actually being very extensively being used as a adjuvant into the relieving the some of the the damages during the malaria, or it is also being used in the drug in another disease which is called as the COPD. Okay, so congenitive pulmonary disease. So N-acetylcysteine is being used in adjuvant therapy. It is a byproduct of glutathione. It is a strong antioxidant. It protects the cells from the oxidative stress. It reduces the inflammation through the reduced secretion of the cytokine, which is called TNF alpha. And NAC thought to be protect the cell by increasing the GSH level inside the cell. So what happened is that during the malaria, you always have the RBC rupture, right? And when there is a RBC rupture, the RBC is actually going to be released the hemoglobin, and that hemoglobin is getting converted into methemoglobin. So hemoglobin, you know that hemoglobin is the oxygen carrier, right? And once the hemoglobin is getting present into the oxygen environment, it get oxidized. So hemoglobin is actually containing the iron as iron 2 plus, right? Whereas when the iron is present as iron 3 plus, it is called as methemoglobin. And because of this conversion, the methemoglobin is very toxic, right? So it actually can cause a lot of toxicity issues, right? And that's what that happens during the malaria. So methemoglobin actually can cause the oxidative stress into the cell, and that's how it is actually going to be kill the cells, right? So since the NAC was antioxidant molecule, we thought that it may be blocking the methemoglobin molecule, and that's how it may be reducing the oxidative stress, and that's how it probably will be promoting the cell survival. So exploring this particular hypothesis, we discovered that the NAC is a suicidal inhibitor for methemoglobin. Now, when we want to do this kind of study, we have to actually take a lot of control experiments. Okay, so for example, we have studied how the because methemoglobin is actually going to cause the RBC lysis, okay, which means it is actually going to. So we first thing what we have measured is we have measured whether the RBC lysis is getting blocked or not. So we did the NAC, GSH, methionine, and NAM. Right, NAM and NAC is having only one difference, that instead of cysteine you have the methionine, and you will see that when I remove the cysteine, right, it is actually not protecting the cell from the hemolysis, right? So there is a hemolysis. Even methionine is also not been able to block, but GSH is been able to block because GSH is doing the same function as the NAC, right? And then the same thing we have done also, that we have found that because the methemoglobin has the peroxidase activity, so it was actually blocking the peroxidase activity also the same way. And then the hemolysis is also being blocked by dose dependently by the NAC. So as you increase the NAC concentration, the hemolysis is going to be blocked. Same is true for the peroxidase activity. So this is true that the NAC is actually protecting the cells from the hemolysis, and NAC is also also inhibiting the peroxidase activity. So then we also asked whether the inhibition is reversible or irreversible. So this is exactly what we have done. And remember, this is the experiment I have just said, right? So what we did is we took the methemoglobin plus NAC. You'll see this is a 100% activity what you got, right? Then we did the methemoglobin plus H2O2, 100% activity. Then we did the methemoglobin plus NAC plus H2, which means this is the complete reaction. Once you did the complete reaction, the activity goes down to 2%, right? And then we took this 100% reaction, we did the overnight dialysis, and there is no recovery of enzyme. This means by doing this, we know that the inhibition is irreversible. Okay, this means the NAC is an irreversible inhibitor of the methemoglobin peroxidase activity. And then we tried the other methods, right? So we tried like protecting it by the substrates and other kinds of things to understand whether the substrate is actually going to protect. So it's clear that if you don't allow the methemoglobin to process the NAC, it is actually going to get blocked, right? This means it is actually going to be a suicidal inhibitor. So if it is a suicidal inhibitor, we also did the suicidal kinetics. So this is the suicidal kinetics. So NAC follows the mechanism based inactivation of methemoglobin. So what you do is in the suicidal kinetics, what you do is you take a reaction mixture and then you are actually going to take out the enzyme at different time intervals, right? Like for example 0.04, 0.08, 0.12 and all that. You allow the inhibitor to react for these many time points, and you will see that the enzyme is fully active, so 100% active. Then it is actually going to show you a reduction in change. Okay, so these are the number of times, minutes, what you have actually kept, and then you can actually be able to calculate the k-ops, and you can be able to calculate the t-inact. So what is the minimum time required to inactivate the enzyme? Okay, and from this you will know that the Ki for this is 0.76, and the affinity of NAC for the enzyme is 8.5 macromolar, and T half inact, which means only 0.9 minute is required for inhibiting the 50% of the enzyme. Then we also did the UV visible spectroscopy to know whether the NAC is being processed by the enzyme. So it's actually being processed by the enzyme, and that's how the NAC is going to be modified by the enzyme, and that's how this modified NAC is actually going to be the culprit for responsible for the blockage. And that's why this was not happening when we were adding the substrate, because if you add the substrate, then enzyme will process the substrate rather than inhibitor. So we identified what kind of modification, right? So what we did is we did an EPR experiment, or electron paramagnetic resonance spectroscopy, and what you found is that there is a sulfur centered free radical what is going to be produced when you are treating the NAC plus methemoglobin plus H2O2. Okay, that is not present when you are removing the hydrogen peroxide, that is not present when you are removing NAC, or when you are adding inactivated methemoglobin, or when you are adding the meth buffer alone. So this was shown that this complete reaction is producing a producing the EPR signal, and that EPR signal is is actually saying that DAC is getting modified. And this what is the modification? Modification is that it is actually forming the sulfur centered radical. Okay, which means some of the cysteine molecules probably have the sulfur where you have the single electrons. So once you have the single electron, it is actually going to go and attack onto the protein's residues, and that's how it is actually going to block some of the crucial residues. So NAC oxidation product is actually interacting with the sub enzyme. So if you add the H2O2, you are actually going to produce the NAC oxidation product, and that's how it is actually going to go and bind. Whereas in you don't have the hydrogen peroxide, it is actually not going to bind. Then what we did is we also identified the what kind of modification. So we found is that the N-acetylcysteine is getting modified, and this modified product is binding onto the protein, and it is actually blocking the hemine. So methemoglobin has the hemine, and it is actually blocking the hemine part. So hemine is NAC is forming an adduct with the enzyme bound hemine to inhibit the peroxidase activity. Then we tested this DAC oxidation product, and what we found is that when you add actually the spin trappers, so these are the single electron trappers. Okay, so if you add the NAC along with the tempo or PBN, so these are the spin trappers actually. So they will actually going to trap the single electron species. Okay, so they are not antioxidant, they are single electron trappers. Okay, so for example, if you have a sulfur centered single electron, they will go and bind. Okay, instead of this allowing this inhibitor to go and bind to the enzyme. So if you have the spin trappers, you will see the hemolysis if the methemoglobin and NAC is present. But if the methemoglobin is NAC is present, you will see that if you add these two, there will be a inhibit. So if you have the methemoglobin, you are going to see 100% hemolysis. When you have the NAC, you are not going to see the hemolysis, but when you have the activity in the presence of tempo or the PBN, you will going to see that the effect of NAC is actually getting reversed. So that actually proves that there is a NAC oxidation product which is responsible for the hemolysis. And ultimately, we have come up with this particular model that NAC is actually having a SH group. So you know that the NAC has a GSH, right? And when it is getting processed by the methemoglobin peroxidase activity with the help of the hydrogen peroxide, NAC is getting converted into the NAC sulfur centered radicals, and these sulfur centered radicals are attacking onto the methemoglobin, and that's how it is actually forming the methemoglobin iron sulfur clusters, right? And this is inactive. So this is actually a inactive enzyme, and that's why it is actually not going to process. If you want to read more about this, you can actually be able to download this particular paper from this particular journal, and it will actually going to tell you the more details about this. So far what we have discussed about the irreversible inhibitions, and we have discussed also about the suicidal inhibitions. So inhibitor, when you develop the inhibitor could be of reversible in nature or the irreversible in nature, and within the irreversible nature you can actually have the suicidal enzymes or suicidal inhibitors, or the mechanism based inhibitors, or the non-mechanism based inhibitor, which means in a non-mechanism based inhibitors, the irreversible inhibitors, the inhibitor will go and modify the active groups onto the active site, whereas in the case of suicidal inhibitors, the enzyme is actually going to process these inhibitors as such like a substrate, and then when it is actually converting the inhibitor into a modified form, that modified form is actually going to have the irreversible binding to the enzyme active site, and that's how it is actually going to inhibit the enzyme. So with this, we are going to conclude our lecture here. In our subsequent lecture, we are going to discuss about the reversible inhibitors, and we are also going to talk about the enzyme inhibition kinetics. So with this, I would like to conclude my lecture. Thank you.